for chapter 2 is organizations of the organisms, the organization of organism. Now without much ado, let's see what it is. Now this is the first part, which is 2.1, the cell structure. Now the cell, first of all, what we're going to do here, we're going to describe a cell, like define what a cell is, and then be able to describe the parts of the animal cells and the plant cells and even the bacterial cells and the function of each part of the cells that we're going to be talking about. So we talk about each part of the cells, each cell, like if you take an animal cell, what it's made up of, and you're going to be talking about what it attains. So we define the cell. Cell is the building block of life. The cell is the building block of life. It's like what makes up the organism itself, because you have to add many cells together to form the organism. So the cell is the building block. Without the cell, we can have the organism. It's the smallest unit. It's the smallest unit of an organism. You can also say the smallest unit or the building block of an organism. That's what the cell is. So if you are asked what is a cell, the cell is the building block of organism. The building block of organism. Okay, that determines every other part of the organisms. Because many cells join together to form the tissue, and many tissues together form the organ, many organs together form the system, and many systems together form the organism itself. So cells are of different types. We're going to look at the plant cells. We study the 5 kingdoms. We study the plant kingdom, the animal kingdom, the fungi kingdom, the prokaryote kingdom, and the protistic kingdom. All of this in chapter 1. Now in this chapter 2, we'll be looking at the basic ones for our IGCSE program. So which is the plant cells, the animal cells, and the bacterial cells. Is that okay? And then in this chapter, we're also going to be looking at the different parts of the cells. Okay? And then we'll compare the structures of the plant cells and the animal cells. So that's what this chapter is all about. Now let's take a look at the first one, which is called the animal cell. The animal cell comprises of these internal parts. It has the cell membrane. So we're going to look at the cell membrane. That's outer covering that covers the animal cell is called the cell membrane, where the water diffuses into the cell, in which water diffuses through to get into the cell. Okay, so we're going to be talking about the animal cell, and in the animal cell we'll be talking about the cell membrane. We talk about the cytoplasm, where the metabolic activity takes place in the animal. We talk about the nucleus, where you have the cell's DNA. The cell's DNA, the organism's DNA are in the nucleus of the cell. We talk about the ribosome, which is site for protein synthesis in the organism. We talk about the mitochondria, which is site for energy release in the organism. Okay, we talk about small vacuole, which contains water for other metabolic activity to take place. Now let's take a look at a second one is the plant cell. We're going to exclusively look at all of this. Okay, we exclusively look at that, but let's take a look at a brief look at the plant cells, which comprise of also the cell membrane, okay, cytoplasm, which we've already described, the nucleus, which we've described, but in addition to the cell membrane, it contains what we call the cell wall. We call it the cellular cell wall. So the plant cells contain the cellular cell wall. This is very vital. Okay, one characteristic that differentiates the animal cell from the plant cell. The animal cell do not contain the cellular cell wall, but the plant cell contains cellular cell wall. So this is very important. The ribosome, like we've described, site for protein synthesis, the mitochondria site for energy release in the organism. Then the chloroplast, another difference between the plant cells and the animal cell, the chloroplast. The chloroplast contains the green pigment, which is called the chlorophyll, that traps the sunlight energy for photosynthesis to take place in the plant. Plant cells are able to manufacture their own food. The plants are able to manufacture their own food. One of the key reasons why they are able to manufacture their own food is the presence of the chloroplast. The chloroplast contains a green pigment called the chlorophyll. The chlorophyll inside the chloroplast are able to trap sunlight energy. They are responsible for the green nature of the plant, and they are also able to trap sunlight energy in order for the plant to make their own food. Animals do not make their own food because they don't have chloroplast. Is that okay? So another thing is the large central vacuum. The plant cells has a very large vacuum, whereas the animal cells has a very small one. We're going to look at this. And then we go to the third type of the cell, that we're looking at in this chapter 2, which is made up of the cell membrane as usual also, which is also made up of cytoplasm, also made up of the cell wall, but this time around not cellular cell wall, it's called marine cell wall. It's made up of marine cell wall, okay? It has cell wall, which makes it look like a plant, and it has a ribosome for photosynthesis. It has a circular DNA, it has a circular DNA, and it has a plasmid, which makes it be, this plasmid gives it the ability for it to be used for genetic engineering, for genetic engineering, be it for mutations and used as for genetic engineering in the bacterial cells. Now, let's move to the next one. Okay, the next one is imagining cells as chemical factories. Okay, cell we say is the building block, okay? So if you look at it, cells are the building blocks of life. Now, cells are like the chemical factories. Each cell is made up of different parts, and each part has a specific function. Okay, each cell is made up of different parts. If you look at a chemical factory, like you see here, you see that it's made up of different parts, and each part has its own specific function in an industry, in a chemical industry. Okay, now let's take a look at how we can study cells and their parts. How we can study cells and their parts. We can use a light microscope or electron microscope. We'll talk about the difference between a light microscope and electron microscope. We use the light microscope and the electron microscope to observe a cell. This is an example of a light microscope. This one that we're showing is an example of what? A light microscope. Okay, you place whatever you want to view, you place it here. The cell you want to view, you place it here. Maybe you use a cutting wood to take a chip, to put inside your mouth, to take a chip, your cheek, and use the cutting wood to touch it, and place it here, and place it here, and then you place it here, and then you can use you can view it from this microscope. This is called a light microscope. A light microscope is able to magnify or make it bigger 1,500 times the original size of whatever you're looking at. It's able to magnify, the light microscope is able to magnify the object 1,500 times the original size that you're looking at, of the cell that you're looking at. But however, the electron microscope is way huge. It's way bigger than that in terms of magnifying or magnification of the specimen cell that we're looking at. Now, the electron microscope is able to magnify the original cell 200,000 times. 200,000 times the original size of the cell in question. So, electron microscope can see beyond boundaries, And this picture depicts the electron microscope telegraphy. Okay? So, let's move to the next one. The next one is how can we study cells and their parts. Now, to study cells, we have to take a micrographs or photographs captured using our microscope. You have to, because you cannot see the cell itself. It's difficult to see the cell itself. So if you want to see the cell, you have to use a microscope and take a micrograph from a microscope or a photomicrograph from a microscope. Either you use a light microscope or you use an electron microscope. So they're able to bring out the picture of all the magnification of the original object and show you the image of it, and we're able to study from there. Then we're able to study the cells and their part. So, for example, if you look at a light micrograph from a light microscope, we see something like this. This is a picture of Salmonella taken from a photomicrograph of a Salmonella, taken with a light microscope. Now, if you take electron microscope and looking at the electron micrograph, the micrograph is the picture you're seeing. Okay, using electron microscope to take this electron micrograph, we can see a black and white images, but they can be artificially colorized. Okay, you can use the electron microscope to colorize it and to show the color of this one. This is the original electron micrograph of the polygrains, while this is the colorized electron micrograph of the same polygrain. So you can see the polygrain, the cell. This is just a cell from a polygrain, and then we're able to label it since we'll be able to see it correctly. Now this is the next one. How can we get a clearer picture of a cell. If you want, you take the plant. If you want a plant cell, if you want an animal, you take the animal cell. If you want a longitudinal section, you have to cut it down, okay, and you get a unit like this. And if you want a transverse section, you cut across the organisms or the cell that you're taking a look at, okay. Now we'll move to the next one. What makes up an animal cell and a plant cell? Okay, originally we've categorized them, we've showed them, right? This first micrograph appears to be like an animal cell, okay, even if the picture looks similar. This one appears to be an animal cell, while this appears to be a plant cell. Okay, we're going to explain that as we move on. So we take the cell and we're able to, which are animal cells and which are plant cells. Now, how can we differentiate between animal cells and plant cells? This differentiation came from the peculiarities, the similarity and the differences in each of the cells. The peculiarity and the similarity and the differences that is found in each of the cells. Like we said, the plant cell has a cellular cell wall, which is not found in the animal cell, and it has a chloroplast, which is not also found in the animal cell. But there are so many other similarities, like the cytoplasm, the nucleus, and so on, the ribosome, the mitochondria, and so on and so forth, that are found in both plant cells and animal cells. Now let's move to the next one. Now the next one says, what makes up an animal cell and a plant cell? What makes up an animal cell and a plant cell? Now let's begin to look at this. You say most cells, they have certain parts in common, right? We've said that. The plant cells, the animal cells, and even the bacteria cells, they have so many things in common. The animal cells has a small vacuum. The plant cells has a very large vacuum. They both have ribosome. They both have nucleus. They both have chromosomes, so on and so forth. They both have cell membrane, mitochondria, cytoplasm. They both have all of this. They both have nuclear membrane. So what is different is the cell wall chloroplast are found in plant cells that are not found in animal cells. Is that okay? So that's that for that. Now let's take a look at what makes up an animal cell and a plant cell. Let's begin to look at what makes up both of them. Okay? What's come up to both of them? What makes up a plant cell, and also make up an animal cell? Number one is the cell membrane. Both plant cells and animal cells, they both have cell membrane. Now, what is the functions and what does it look like? It is partially permeable. For example, if you take a potato, you peel your potato, and you use a knife to create like a hole in the potato. If you pour water in the hole in the potato and leave it for some hours, you see that some of the water would have drifted out of the potato and come out to the plate where you put the potato, because it's called partially permeable. Not only potato, like a yam in the case of African scenario. They have a lot of yam. So you peel the yam, you create a hole in the yam, pour water in the hole. After some hour, the water will get out of the yam and gets to the plate where you put the yam. This is because it's partially permeable. So partial permeability is the ability for osmosis to take place within that context. The ability for osmotic reaction or relationship to take place within the system. So when we say a cell membrane is partially permeable or semi-permeable, it means that it will allow the movement of water over a period of time. It will allow the movement of water from region of higher concentration to region of lower concentration over a period of time. Now, the function is that what does cell membrane do for the plant cells or the animal cell? It controls substances that are entering and leaving the cells. So its major job, this is the cell membrane, this one. The line that creates the circle. The line that looks like the circumference. It's not pure circle, but the line that looks like the circumference is called the cell membrane. Look at it. This one is the cell membrane. This is the cell membrane. Now this cell membrane, what does it do? We say it controls what enters the cell and what goes out of the cell. It's responsible, is accountable to tell or to permit what to go in and what goes out of this cell. That's the major jobs. Now let's look at the cytoplasm. Most cell activity occur in the cytoplasm. Most of the activity like metabolism, most of the activities that are taking place in the cell, they take place in the cytoplasm. They take place in where? In the cytoplasm of the cell. Most of the activity happening in the cell takes place in the cytoplasm. Now, we say the cytoplasm, the they contain very tiny dots. All these tiny dots you see, all this tiny structure or whatever you want to call them, all this tiny structure you see in the cell, they are called the cytoplasm. Okay, they contain many tiny structures that are specialized for particular function. They specialize for specific function. Okay, some are for reproductive action, some are for thinking action, some are for sweating action, some are for homeostasis, some are for... So these cytoplasms, they carry out the metabolic activity like digestions, like breathing, digestion, respiration. Okay, all of this functionality that happens to cells are carried out in the cytoplastic reticulum of the organism. Is that okay? So that's their job. They are responsible for metabolic activity or contains the special functionality of the cells, special functionality. And let's go to the third one, which is also common to both plant cells and animal cells, which is called the nucleus. Now the nucleus, it controls the activities such as the growth, the cell growth, and the repair of any damaged tissue, any damaged part of the plant. So the nucleus controls the growth and the repair of any damaged part, and it's also needed for cell division. Because of the DNA, this is where you have the cell DNA. The DNA is found in the nucleus, and hence, since it contains the cell's DNA, it therefore means it controls the growth. Controls growth, repair of damaged tissue, and cell division. Okay, now we go to the next one. The next one is what makes up an animal cell and a plant cell. What makes up an animal cells and a plant cell? A plant cell also has a cell wall besides cell membrane, but animal cells don't have a cell wall. Now, cell wall is that thick, the thick one, the stem of a plant. When you hit it with a cutlass, the stem, that thick outer covering of the plant, the thick one of the stem, is called the cell wall. It's called the cell wall. Okay, it encloses the entire cell. So this one, from diagram, this whole thing is the cell wall. This one is the cell wall. It covers not the inner one. The inner one is the cell membrane. Okay, is the cell membrane. This outer one, the outer one is the cell wall. Okay, together with this thick one, okay, is the cell wall. Okay, this is the cell wall. It's covering the entire plant cells, and the cell wall is fully permeable, and it's made mainly of cellulose. It's made mainly of cellulose for storage of starch and so on and so forth. It protects the cell from injuries and gives the plants their shape and the fish shape. The fish shape you see the plant cells have, it is this cell wall that gives that rigid and fixed shape to the plant. The cell wall, this cell wall is responsible for the cell shape, rigid cell shape, plant cell shape that you see around, and also protect the plant from injury because it's very thick. It's very, very thick, so it protects the plants from injury, protects it from injury. Is that okay? And another thing, like I said earlier, is the chloroplast. The chloroplast that is found which contains the chlorophyll. The chlorophyll is the green pigment that traps the sunlight energy for photosynthesis to take place in the plant. For photosynthesis to take this chloroplast. So the manufacturing of this takes place in this chloroplast. Okay? So the rest you see are also found in the animal cell. But these two are the main distinctions between the plant cells and the animal cell. Now let's move to the next one. Now what are the structures? What are the structures in the cytoplasm? What are the structures in the... Okay, now in the cytoplasm we found things like the ribosome, the mitochondria, the chloroplast, and the vacuoles are all examples of tiny structures in the cytoplasm. They are all found in the cytoplasm or around the cytoplasm. Okay, the ribosome, which is the site for protein release. This one's mitochondria, site for energy release. Chloroplast, which contains the chlorophyll, the green, which makes the green pigment that traps the sunlight energy. The vacuole, which contains water for the metabolic activities in the plants, are all examples of structures in the cytoplasm. So what are found in the cytoplasm, we can mention the ribosome, the mitochondria, the chloroplast, the vacuole, and so on and so forth. Okay, so these are the cell membrane. This line, the outer line, are usually and always the cell membrane. okay, which covers the cell, which controls what enters and what goes out of the cell. The mitochondria, I've already explained this one. Mitochondria is the site for energy released during respiration, and vacuole contains water for metabolic reactions or activities. And then we have the cytoplasm, which is where the activities, metabolic activities are actually taking place. And then we have the ribosome attached to the membrane also in this place. You can see the ribosomes are attached to the membrane and the nucleus and so on and so forth. This is the cell nucleus. These are the cell nucleus. Okay, so this is what it is. Now let's move to the next one. What are the structures in the cytoplasm? Now, what are the structures in the cytoplasm? Now, let's start with the chloroplast. Chloroplasts are absent in animal cells. We don't have chloroplasts in animal cells. Okay? Like we said already, they are only found in plant cells and they contain the green pigment, which traps the sunlight energy for photosynthesis to take place. Now, they contain the chlorophyll. The dark green pigment is called chlorophyll. Chloroplasts contain chlorophyll. You know, you have to understand this. Chloroplasts contain chlorophyll. Chloroplasts contain chlorophyll that are essential for photosynthesis in plants. Is that okay? This is a zoom of it. We just take a stack of it and zoom it out. Stack of the membranes of containing the chlorophyll. Okay, this is just a stack. The whole of this is chloroplast. So we just take a stack of the membrane of chlorophyll, which is this green pigment, responsible for trapping sunlight energy and responsible for the manufacture of food. Now we talk about the vacuole. Then another one in the plant, in the plant cell, are the fluid spaces that stores water. They store liquid, okay, within the cell. Now plant cells, they have a very large central vacuole. The animal cell has a very small vacuole. The plant cell have a very large central vacuole that contains the liquid called the cell sap. Contains the liquid called the cell sap. Here you have water. Animal cells have small vacuole called the vesicles. The animal cell contains the very small vacuole, which we also call the vesicles, that contains water and food substances. It contains water and nutrient. You can say water and nutrient in animal cells, what you call the food substances. Okay, this is what it is, the vesicle. Actually, the vacuole in animal is called the vesicle. So you can call it vacuole or vesicle. Okay, this is what it looks like. It contains water and nutrient. Now we go to the next one. Okay, here is a question. Let's investigate. It says compare the epidermal cells with the diagram on student book. Okay, so this is what you're to do from your textbook. Okay, now let's move to what makes up a bacterial cell. Now we've talked about the plant cells, the animal cells, their similarity and their differences. I think we only itemize two major differences, which is the chloroplast, excuse me, which is the chloroplast and the cell wall, cellular cell wall, for the between plants and animal cells. Now for bacterial cell, let's see what it contains. First of all, a bacteria is a unicellular organism. Plant and animal are multicellular organisms. We forgot to mention that, but we're mentioning it down now. But I think we did it in chapter 1. The plants and the animal cells, they are multicellular organisms. The plants and the animal cells, they are multicellular. But bacterial cell is a unicellular, meaning it contains only one cell, only one type of cell. Multicellular meaning it has multiple cells, okay? Multiple types of cells. Okay, now, bacteria is a unicellular, that is made up of only one cell. It's one cell organism only, okay? So what it contains a cell membrane. A bacterial cell contains a cell membrane, which makes it look like plants and animal. It contains cytoplasm, which also makes it look like plants and animal. It contains a large circular DNA. In plants and animal, we say they contain nucleus, which contains the DNA. But this one we simply say, the bacterial cell, we simply say it contains a circular DNA. Contains what? A circular DNA. And then it also contains one or more small circles of DNA called the plasmid. It has small circles of DNA called the plasmid. Small circles of DNA. This is the major circular DNA. This is the major circular DNA in a bacteria. So if you ask to draw a bacteria, you draw something like a plant cell or something like animal cell, you draw a huge DNA and you draw small DNA. These smaller unit DNAs, they are called the plasmid. And this plasmid makes it to be able to be used for biotechnology application. Remember this. This will always come in multiple choice exam. Okay, it will always come in multiple choice exam. Which of the following makes a bacteria to be used for biotechnology? The plasmid or plasmidity, however they say. Then the bacteria cell, apart from circular DNA, cell membrane and circular DNA, it contains also small tiny DT called the ribosome. It contains the ribosome, it contains the cell wall, okay? It contains this outer layer that makes it look like a plant called the cell wall, okay? And now it also contains the cell membrane, the innermost membrane, okay? And then it contains the cytoplasm, which is the whole yellowish coloration, called the cytoplasm. And then it contains the flagellum, which enables it to swim. The flagellum is the tail that enables it to swim. Flagellum is the tail that enables it to swim. Okay, so that's that for that. Let's move to the next one. Now, how are animal, plants, and bacteria cells similar and different? They're similar. What's the similarity between plants and animal cells? So the similarity, you see, we rule this table up to only to this level. To show there are differences between plant and animal cell. And therefore, similarity, we don't break the table. We leave it. What is similar between them? We did continue. Whatever is similar, we write it together. Like is part of the plant cell and also part of the animal cell. Okay? Now, what is different? First of all, animal cells do not have a cell wall. That's number one difference between plant and. Whereas the plant cells, they have what we call this cell wall made of cellulose, or you can simply say cellular cell wall. The plant cells have a cellular cell wall. The animal cells do not have a cell wall. That's number one difference. The bacterial cells, they have a cell wall made of murein, not cellulose. They have a cell wall made of murein, which is not cellulose. So that's the only difference. Then another difference between them is that animal cells do not have a chloroplast. I have itemized this a couple of times. Whereas the plant cells, they do have a chloroplast. The third difference is that they have many small vacuum. That's the animal cells have multiple small vacuums, whereas the animal cells have a very large central vacuum. So these are the three differences between them. But even though they both have vacuum, the animal cells have multiple small vacuums, and while the animal cell has a large central vacuum. So these are the three differences in the exam. If you have to make differences between the plant cells and the animal cell, you give these three differences. Now, in the case of bacterial cells, they do not have a chloroplast. So just now, because they have a cell wall, it looks like as if they are plants, but they don't have chloroplast. So which makes them not look like plants anymore. Now, they also have some may have large vacuums. Some of them have large vacuums, whereas some do not. Now let's take a look at the similarity between the plant cell and animals. So they have nucleus, enclosed by nucleus membrane. Both plants and animal cells, they have nucleus that are covered by nucleus membrane, and they both have membrane-bound structure, like the mitochondria. They both have the mitochondria, both the plant cell and the animal cell. They both have DNA. It's both linear. They both have DNA in them in the nucleus, and they do not have plasmid. Both of them do not have plasmid. Both the plant cell and animal cell. This is only found in the plasmid, which makes it to be used for biotechnology or bioengineering, is because the bacteria have plasmid. And then the DNA here is circular. They have a circular DNA, circular DNA, and they have smaller ones, which makes up the plasmid. Okay. And they do not have cell membrane. They do not have a cell membrane, or the one that is bound one. They don't have the bound cell membrane. So these are the differences and similarity between the three. Okay. Here is a practice question. State the functions of this cell part: the mitochondria, the chloroplast, the ribosome. So you pause the video and and answer this question. Okay, the vacuum. You're able to state the functions of this part, what they do, each of this part, and state and explain three differences between the plant cells and the animal cells. Okay, so you pause the video and answer those questions. Then we move on to 2.2. We're done with 2.1. Now here we go to the levels of organizations, levels of organization in the plant's arrangement. Now in this section, we're going to state that new cells are produced through cell division. New cells, cells, they divide to make new cells. That's growth. When cells divide, they make new cells. Cells divide, they make new. Then you'll be growing and be bigger and bigger. So we're going to talk about cell division a little bit, and we're going to state that specialized cells perform specific function. Every specialized cell does that specific function that it has been designated to do. Then here we also describe the term tissues, organ system, and the organisms. So now cells, when we organize many cells, we put many cells together, or many specialized cells. When we put them together, they will form what we call the tissue. And when we put many tissues together and organize them together, many of them, they will form what we call the organ, or what we call the organ. Then, if you put many organs together, they will form what we call the organ system, or what some textbooks will just call systems, like the respiratory system, digestive system, hormonal system, circulatory system, and so on and so forth, skeletal system, and so on and so forth, muscular system, and so on and so forth. So many organs put together. A good example of organ is the heart. The heart is an organ. The muscles, they are organs. So if you put like your nose, they are organs. Your mouth, an organ. So you put many organs together, they'll form the organ system, like the digestive system, respiratory system, and so on and so forth. And then if you put many systems together, you get the whole organism. To form many systems. You take the respiratory system, digestive system, hormonal system, coordinatory system, circulatory system, skeletal system. If you put all of them together, you form the organism. So now let's move to the next one. Now, how is the structure of a cell related to its function? How is the structure of a cell? How does it relate to its function? Yes, the creator of everything has so much function that the cell to be able to look the way it looks in order to be able to carry out the specific function that is meant for it to do. New cells are produced through cell division. Okay? Existing cells, we multiply, we divide, we grow, swell up, and divide into 2 or 4, depending on the division case. Now, differentiation is the process by which a cell becomes specialized for a specific function. A cell becomes specialized for a specific function. There are cells responsible for respiration. There are cells responsible for reproduction. There are cells responsible for any metabolic activity, sensitivity, and all of coordination, and all of these things. There are cells responsible to detect stimuli, external stimuli. Okay, so every cell has its own specific function. For example, let's take a look at the palisade mesophyll cells. They are used for photosynthesis. The palisade mesophyll cells, they are used for photosynthesis. This is where you have the chloroplast and the nucleus. Okay, the palisade mesophyll cells in plant cells is where photosynthesis takes place. Okay, they contain chloroplast, which contain chlorophyll, which traps the sunlight energy in order to activate the water and the carbon dioxide to react together to make glucose. Okay. Now another type of cell is the root hair cells, which is found under the ground, which helps to absorb water through osmotic process, and also to absorb water and mineral ions that will eventually enter the xylem cells to take all this water and mineral ions to the rest of the body of the plant. So different cells perform specific function. Now let's move on to the next page. How is the structure of the cell related to its function? How is the structure of the cell related to its function? Now, for example, let's say the red blood cells, they are used to transport oxygen. You can see their shape, the surface view, and this is the cytoplasm containing the hemoglobin. Okay, the cell cut into two to show its biconcave shape. We call it to two to show its biconcave shape here. So they carry oxygen around the body, around to every other part of the cell. Here is the egg and sperm cells. This is the sperm cells. This is the egg cell. It has a jelly coat that covers the egg. It has a nucleus inside. It has a cytoplasm. Okay, everything has cytoplasm, tiny dot. Okay, has the cytoplasm. This is the egg cell, and this is the sperm cells. It has the cytoplasm. It has the nucleus here, and this is called the head. The head is sometimes in biology exam they ask you to draw this sperm cell. You have the head, which contains the nucleus and the cytoplasm, and then you have somewhere in the middle where the mitochondria is, the middle piece, and then you have the tail, which is the flagella, which enables the sperm cell to swim to the egg cells. Then you have the ciliated cells for movement of mucus in the trachea or the bronchi in the respiratory system. The ciliated cells for the movement of mucus in trachea and bronchi. Is that okay? And then we have the nerve cells during the nervous system. The nerve cells, or what you call the neurons, for the transmission of electrical impulses to the brain. This is a neuron, nerve cells. Okay, now how are cells organized to form an organism? Many cells will join together to form tissue. A tissue is a group of cells that work, a group of cells with similar structures that work together to perform specific function. Example is the muscle tissue. Our muscle is a tissue. Okay, a muscle tissue is made up of tissue of muscle cells, and we have the leaf epidermis, which is made up in the case of plant cells, the leaf epidermis is made up of epidermis cells, okay, which is a tissue as well. Now we move to the next one. How are cells organized to form an organism? Still on that. Many tissues will come together to form what? Many tissues will come together to form organs. Many tissues will join together to form organs. So what is an organ? An organ consists of more than one kind of tissue. Working together to perform a specific function. So organ contain more than one tissue that works together to perform a specific designated function. Okay? A good example, the stomach contains glad tissues that forms the organ, muscle tissues and nerve tissues. Okay? These tissues, they work together for the sake of the digestion of food. Whether the glad tissue in the stomach or the muscle tissue around the stomach or the nerve tissues, they all work together for the digestion of food. The leaf contains mesophyll tissue, the xylem tissues, and the phloem tissue. Okay, all of these, they work together for the plant nutrients and transport. They all work together in mesophyll tissue, xylem tissue, and phloem tissue for transportation of manufactured food, especially the phloem for transportation of the manufactured food, the xylem for transportation of water and mineral ions for the manufacture of food, and so on and so forth. Okay, now let's move on. How are cells organized to form an organism? For cells to form an organism, you have to put many organs together to form the organ system. Hence, the organ system is defined or is made up of several organs working together to perform same purpose, to perform common purpose, to perform similar purpose. For example, you put many organs together to form, like the trachea is an organ. Okay, you bring the trachea here. Look at the trachea, you bring it here. You bring the lungs, they are also organs. You bring these two lungs here, they are organs. They join together to form what? The respiratory system. They will join together to form the respiratory system. Okay? So you have the stomach is an organ. The stomach is an organ. The intestine, the small intestine and the large intestine, they are each individual. Each of them, they are organs. The liver, whatever you want to call them, they are organs. Then you bring that together, both the stomach, the small intestine, the large intestine, the liver, the pancreas, all of them, you bring them together to form the digestive system. they form the digestive system. Okay, so you can see when you put many systems together, organ systems together, they will form the whole organism. You have the one for coordination, the brain and the neurons and everything. You have the respiratory system here. You have the digestive system here. You have the muscular system and the skeletal system. Everything put together will form the whole organism. Everything put together will form the whole organism. So organism is starting from cell to tissue, tissue to organ to organ system. Then before organisms are formed. Now let's take a look at 2.3, size of specimen. Let's take a look at the size of a specimen. Cells are small. Many cells put together forms tissue. Many tissues together form organ. Many organs together form organ system. Many organ systems together form organisms. Now in this section we're going to state and use the formula of magnification. Okay, we want to carry out magnification. This is always in the exam, multiple choice exam, and even theoretical exam in biology in IGCSE. So you need to know how to do calculation in magnification. Here we will study magnification of cells, of organs, of tissues, of organs, of system, and so on and so forth. Then we're going to calculate magnification and the size of the biological specimen in millimeters, and also do the conversions of measurement from millimeter to micrometers, and so on and so forth. Okay, so cells and biological specimen can be measured in millimeters or micrometers. You can measure them in millimeters or micrometer. Micrometer means times 10 to the power of -6. When we say micrometer, means times 10 to the power of -6. When you say millimeter, means times 10. When you say micrometer, say times 10 to the power of -6. This is micro. Okay? The symbol is this. Micro is like this. This is the symbol. Okay, micro. So when I say micrometer, I do like this. So when I say millimeter, I will do times 10 to the power of -3. Millimeter is times 10 to the power of -3. And it is millimeter. Okay, millimeter. Okay, now we say cells can be viewed under a microscope. And when you view it under a microscope, magnification can be calculated from the image or from the drawing, from either the image or the drawing. And the formula for magnification is simply saying magnification is the size of the image over the actual size of the specimen. Size of image or your size of your drawing over the actual size of a specimen. So magnification M is equals to size of image. So I can say image size over actual size. You can say image size over actual size or drawing size over actual size of specimen over actual size. Okay, magnification is simply this formula. Okay, now let's move on. Now let's take a look at an example. How do we calculate magnification? How do we calculate? We already state what it is in ratio. We say magnification refers to how many times the image of a specimen is enlarged. How many times it has been enlarged. You know, when we talk about light microscope, we say it can enlarge the image 1,500 times. We say it can enlarge, light microscope can enlarge it 1,500 times. But electron microscope can do it 200,000 times. Okay, electron microscope can do it 200,000 times. Okay, so magnification simply means how many times the image of a specimen can be enlarged. So in case they ask you to define magnification, it tells the number of times the image of the object is enlarged. And the formula is the size of the image or drawing over the actual size of the specimen. Now let's take a look at these tomatoes. For example, a tomatoes, we cut a sectional cut of a tomatoes fruit. Now we say figure 2.7 shows the transverse. This is a transverse section because we cut it horizontally. It's a transverse section of a tomato fruit, right? A transverse section of a tomato's fruit. Now the actual diameter of the tomato fruit is 50 millimeter. The actual, remember the formula, actual will be at the bottom in the formula. We say magnification is the drawing, or either you say drawing or image size over actual size, over actual size, over actual size. Now, you said the actual size is 50 millimeter. So I'll have 50 at the bottom, 50, because it's for actual. Calculate the magnification of the tomato fruit. Show your working. Okay? Show your working. Now, the diameter is shown here. This is the diameter. So you can measure it. Okay? You can measure it. So when we measure it, probably maybe we'll go 40 millimeter. We'll go 40 millimeter. So you have to measure it. See if it's not told to us. So you say you get, when you do the drawing measurement, you get the 40. So you say image size, which is 40, over actual size, which is 50. You divide, you get 0.8. So the magnification is 0.8. 0.8 shows that it's going to be smaller. Okay, means it's going to be smaller than the original one, the actual one. Okay, we move to, you say, how can we convert between micrometer and millimeter? I already showed you that. So a millimeter is times 10 to the power of negative 3, or 1 over 1,000. If you want to change a millimeter to meter, you multiply by 10 to the power of negative 3. Or you multiply by 1 over 1,000. 10 to the power of negative 3 is same as 1 over 1,000. Same as 1 over 1,000. Now, for micro, you multiply by 10 to the power of negative 6, which is 1 over 1 million. 10 to the power of negative 6. Whatever they give you, you multiply by 10 to the power of negative 6, which is same as 1 over 1 million. 1 over 1 million. That's 60. 1 over 1 million. Okay? So that's that for micro. Now let's move. These are practice questions for the students. Here, they complete the table to describe how the cells have been adapted to carry out their function. Okay? How the cells has been adapted to carry out their function. Say function to absorb sunlight for photosynthesis. How is it adapted? You state the important characteristics that serve that function. Contain chlorophyll, plast, which contain chlorophyll that traps sunlight for photosynthesis. So you state all of this. Okay, you answer this question. Let's move on quickly. Okay, so I think we are almost in the end of this one. So in summary, what we have for this class is that we've taken a look at the cell, the plant cells, the animal cells. We studied them, and the bacteria cells, and we looked at the organizations of cells, and we looked at the functionality of each part, and so on and so forth. So with this, we say we've come to the end of today's class.